All right, today we're jumping into two really fascinating worlds, biology and programming. We're going to talk about an amazing tool called BioPython, which is literally helping scientists decode the language of life itself. So let's dive right in. So let's kick things off with a problem that is, frankly, mind-boggling. The human genome. It's like a book with 3 billion letters. How on earth do you even start to read that? And more importantly, how do you find the tiny typos that could lead to disease when you have to compare it to thousands of other genomes?
So here's our game plan. We'll start with that massive data challenge, then introduce our solution, BioPython. From there, we'll walk through exactly how it decodes DNA, searches through the entire library of life, and even helps us visualize molecules in 3D. You know, the core issue here is what we call a data deluge. Every single day, labs all over the world are generating a tsunami of information. We're talking DNA, RNA, protein sequences, and it's all flooding into scientific databases. It's impossible for any one person to keep up. And just to put that into perspective, a single database, GenBank, already has more than 200 million sequences. Trying to find one specific needle in that massive haystack? Yeah, that's going to require some serious computational firepower. And this is where the hero of our story comes in, BioPython. Honestly, the best way to think about it is as a computational Swiss Army knife for biologists. It's this one super versatile toolkit that can handle an enormous range of tasks.
Officially, BioPython is this collaborative open source project. And what's so cool about that is it's built by scientists for scientists. It provides free, accessible tools that absolutely anyone can use to start tackling these huge biological data sets.
So what's actually in the Swiss Army knife? Well, with BioPython, you can read all those weird, specialized biology files. You can connect to massive online databases, run super powerful search tools like BLAST, and analyze everything from a simple DNA sequence all the way to a complex 3D protein structure. Its versatility is really its superpower. Okay, so let's put this thing to work. Imagine we just have a raw DNA sequence. It's just a long string of letters. How do we figure out what it actually does? That's the first puzzle that BioPython helps us solve. And this is the first kind of brilliant step. BioPython doesn't just see Gattaca as a bunch of random text. No, it creates something called a seq object. And that object basically tells the program, hey, heads up, these aren't just letters, they're DNA bases and they follow specific biological rules. All of a sudden, our text has context. Then you've got the seq record object, which I love to think of as a library card for that sequence. It holds the sequence itself, yeah, but also all of that crucial metadata, its unique ID, its name, a description of what it is. Now our data isn't just data, it's organized and meaningful. And now, with this organized data, we can do something pretty incredible. We can digitally replicate one of the most fundamental processes in all of biology, what's called the central dogma. We can take our DNA, transcribe it into RNA, and then translate that into a protein, all with just a few lines of code. And the process itself is astonishingly simple. You take your DNA seq object, and you run a single command, .transcribe. And just like that, BioPython instantly converts it into messenger RNA, swapping the DNA base T for a U, exactly like a real cell would do. It's amazing. The next step is just as powerful. We take that messenger RNA we just made, run the .translate command, and bam, out come the string of amino acids. These are the building blocks of a protein. So in just 2 simple steps, we've gone from a genetic code to a molecule that has a potential job to do in a cell. So we have a protein sequence, but what is it? You know, a sequence all by itself is kind of like a word without a dictionary. To understand what it means, we need to compare it to all the other known sequences out there in the world. This is where a tool called BLAST comes in. Think of it as the Google for biology. And BioPython makes using it incredibly easy. You can write a really simple script to take your sequence, send it off to query the entire online NCBI database, and then get the results delivered right back to you to analyze. And BioPython neatly organizes all those results for you. Now, the most important column to look at here is the E-value. You can think of it as a coincidence score. The lower the number, the less likely it is that the match is just random chance. It means you've probably found a true biological relative, which gives you huge clues about what your protein might do. But life isn't a one-dimensional string of letters, right? Proteins are these complex, folded 3D machines that do all the work in the cell. So how do we get from our simple sequence to its actual physical shape? Well, to solve this, BioPython has a module called bio.pdb. It's specifically built to read these complex 3D structural files from the global protein databank. This allows us to stop thinking about molecules as just strings of letters and start actually working with them as physical objects. The real genius here is how it's organized. BioPython arranges all that 3D data in this really logical hierarchy, kind of like nested folders. You have the main structure, which contains models, which contain chains of amino acids, all the way down to individual atoms. This lets you zoom in and out and navigate a super complex molecule with incredible ease. And this organization lets you do some really powerful analysis. For instance, you can pinpoint any two atoms in the entire protein and calculate the precise distance between them in angstroms, which are, by the way, incredibly tiny. You're asking real-world physical questions about a digital object. It's pretty cool. And the usefulness just keeps going. I mean, biological data comes in dozens of complicated, often really messy formats. BioPython can just clean it up for you. With one single command, you can convert a dense, hard-to-read GenBank file into a simple, clean, FASTA format. It makes a researcher's life so much easier. So, when you really get down to it, BioPython is more than just a tool. It's a new language for asking biological questions. It empowers anyone to read the book of life and find stories that have never been told before. The only real question left is, what story would you ask it to tell?